Andrews. And I want to say that back almost 20 years ago, Cal Harley and I, working together at Geron Corporation, discovered human telomerase, which has made a really big difference. One of the things I liked about being at Geron Corporation is we made the information available right away to the world. Suddenly thousands and thousands of publications came out that really enhanced this whole field, letting everybody know the real power of telomeres. I'm going to be talking a little bit about that later. As a result, we were awarded second place for National Inventor of the Year for discovering human telomerase. I've been a telomere biologist for 19 years now. I've been in biotech for 31 years. Before doing anything with telomeres and telomerase, I was doing cancer research, cardiovascular research, and inflammation research. I'm presently on 43 U.S. issued patents on telomerase, and as Dean was just saying, I'm the founder, president, and CEO of Sierra Sciences. I've also been recently in popular science magazines, The Today Show, etc. So this is a testimony of the fact that this field is actually really blossoming a lot more than it did 10, 20 years ago. So kind of like my message that I always say is that bad things happen when telomeres get short. There's really nothing wrong with long telomeres. But I'm going to just focus on the fact that telomeres wreak havoc inside of all of us. And so I know this is going to be a little redundant with what Noel's already showed. Great video, Dean. I guess you put that together. But what is a telomere? And so to answer that question, and most of you probably already know, but let's zoom in on a human being. And we see that a human is made up of 100 trillion cells. And most of the theories on why we age say that we age because our cells age. So if we could figure out a way to stop the aging in our cells, that would be the way to stop aging in ourselves. But let's zoom in further. And of course, every cell has a nucleus. And in the nucleus, there are found these chromosomes where all the genes are. But if we zoom in any further, we see that on the very tips of the chromosomes are found the telomeres. And you know, chromosomes, the DNA is kind of wound up in a chromosome like a slinky from one end to the other. And at the very ends, if we unravel that slinky that's part of the telomere, we find that when a telomere is typically about 15,000 bases in length, DNA is measured in units called bases, it's about 15,000 bases in length, at least it is when we're first conceived. So as soon as our cells start to divide, our telomeres start to shorten. And by the time we are a newborn baby, our telomeres have shortened from about 15,000 bases down to about 10,000 bases. This is a measurement that's been repeated over and over again. Telomeres shorten as cells divide. Okay, as the baby starts to develop further, the telomeres get shorter and shorter and shorter until they get down to about 5,000 bases, and then cells lose the ability to function when telomeres get as short as 5,000 bases. And as a result, that's when most people die of old age. Let me go over that again. We're conceived at 15,000 bases, we're born at 10,000 bases, and we die of old age at about 5,000 bases. And there's absolutely nothing we can do about this yet. No matter how much you eat, no matter how much you exercise, no matter how much you try to eat the perfect diet, live the perfect life, you cannot stop this shortening. Now, there's a theoretical maximum that people have published on several different papers saying that we have a theoretical maximum lifespan of about 125 years. But nobody's ever been able to explain that before. But the shortening of telomeres fairly accurately explains this 125-year theoretical maximum. And so this is one of the reasons why telomeres have become such a focus as being a real possible explanation of aging and more than just aging. So, as Noel already said, the Nobel Prize in Medicine was awarded to Elizabeth Blackburn, Carol Greider, and Jack Szostak for doing the pioneer work that led up to all the work that's going on right now, characterizing telomeres, showing that telomeres had something to do with cellular health and that short telomeres were bad for you. So I want to just congratulate them for that. Next, I want to just summarize. I've talked about the fact that telomeres shorten, but why do they shorten? Okay, and so what I try to do is use an analogy here of a chromosome being the same as a top layer of bricks on a brick wall. And so when a cell divides, the chromosome needs to be duplicated or replicated so that the two new daughter cells have the identical chromosomes. And so I kind of look at the replication as laying a new layer of bricks on top of that brick wall. So let's go ahead. We got this brick layer inside the cell. And of course, he's called DNA polymerase I inside of a cell. And he's laying the bricks. And this is a long, tedious process. Chromosomes are about 100 million bases in length. So it's going to take a long time. And I'm just going to go skip over here to the telomere. And so what we want to see is really what's happening at the telomere. And so when the brick layer gets to there, you're going to see that there's a little bit of a problem. Because of the fact that he's kind of on top of this wall, as a result, the brick layer cannot lay a brick in the last place the brick layer was standing. As a result, the new chromosome is shorter than the old one. Now this is surprisingly very similar to what's happening inside of a cell. Okay, so again, the brick layer is going to, so the cell wants to divide again. The DNA needs to replicate again. The chromosome is going to get shorter again. And on and on and on. And again, no matter how well you eat, no matter how much you exercise, no matter how much you do everything that you can think of that's healthy, you can't stop this type of shortening. Okay, but there are things that you can do to accelerate the shortening and deaccelerating, as Louisa would be talking about later. Anything related to an unhealthy lifestyle that includes like obesity, lack of exercise, psychological stress, and smoking. All of these things will cause accelerated telomere, shortening of the telomeres by generating free radicals or causing inflammation, which will cause the telomeres to get shorter at a fast rate. And that's how sometimes we see people getting accelerated aging, or we see diseases that are often associated with accelerating aging diseases. But you can do something about this. You can exercise, you can lose weight, you can quit smoking, and you can meditate. And a lot of these things Louisa would be talking about, you can do to help decrease the rate of accelerated aging. Now I call this accelerated telomere shortening. And the type that I described before that you can't do anything about, which is just a part of the lack of ability of the cell to replicate the end of the chromosome, I call that basal level telomere shortening. And there's nothing we can do about that right now. Okay, so the best example that I know of that shows that telomeres have something to do with aging is that some people are born with short telomeres. And this disease, called progeria, is kind of a disease of short telomeres. And these kids have this life expectancy of about 20 years. And it's a horrible disease. They suffer from all the same kind of diseases that normal old people do. And if we could find a way to prevent telomere shortening, this could be a cure for these kids, and along with a whole lot of other things. And so remember I started off with saying bad things happen when telomeres get short. And this is one of the best examples of a bad thing that happens when telomeres get short. We really don't want bad telomeres, short telomeres. We don't want bad ones either. But it's not just these kids that are suffering. It's now because of all the publications and all the people all over the world that have been looking at telomeres, and especially with the use of the enzyme telomerase. There's been found to be practically every disease you can imagine has been shown to have a connection to short telomeres. That's cardiovascular disease, cancer. Even at Geron Corporation, when I was still there, some of the scientists there were looking at the endothelial cells that line the blood vessels and showing that areas around plaque had shorter telomeres than areas that were not surrounded by plaque. And so it was suggesting that telomeres played a role in cardiovascular disease. Short telomeres can cause it, long telomeres don't. And there's been probably 100 publications since supporting that. But same with cancer. Cancer has been now shown, lots of publications recently have shown that when telomeres get short, you get a high rate of chromosome rearrangements, mutations that can lead to cancer. And so again, keeping your telomeres long is a way to decrease your risk of getting cancer. Also, shortened telomeres would decrease your ability of your immune system to fight diseases, including cancer. And so you want to keep the telomeres long in your immune cells. So it's just a lot of different things. Look over this list. If anybody wants to talk about anything in particular here, because I don't have time to go over all these today, I'd be happy to discuss any of these with you, along with all the other peers that I have here today. So again, this is an example that bad things happen when telomeres get short. And as I said, it's not just aging. It used to, years ago when I got into this field, that's what I was interested in. I was trying to find a way of curing aging. But now I'm finding out that telomeres play a role in everything that we do, and it's not just aging. So I'm calling this now telomere shortening disease. So instead of me going around saying I'm trying to cure aging anymore, I'm going to be saying I'm curing telomere shortening disease, because it's the #1 health risk that I think we all have. And one of the problems with this is that since we all have it, we all consider this normal. But in 100 years from now, when this problem doesn't exist and somebody has this telomere shortening disease, everybody's going to think, boy, this person needs medical attention. But that's because it's not going to be normal then. But right now it is normal, and so we all just think this is normal. We should be growing old. I don't believe that. So back in the 60s, Nixon declared war on cancer. And not everybody suffers from cancer, but everybody does suffer from short telomere disease. And so I think it's time that we just start saying we need to have a war on telomere shortening. And as far as I'm concerned, I am a very active participant in this war. I am really trying to figure out a way to prevent telomere shortening. And to get started, the governor of Nevada just declared, just in a proclamation, just declared that September 2nd to 8th is now Telomere Research Week in the state of Nevada. And we'll see if that follows suit elsewhere. But I think we're on to a good start. And so what I want to do right now is, what can we do about telomere shortening? And the first thing that I want to talk about is that it's important to know the length of your telomeres. There are techniques out now. A lot of people say, well, why do you want to know the length if there's nothing you can do about it? But there are things you can do about it, and Louisa will be talking about some of those a little bit later. But to measure the telomeres, you typically, people, if you look at this graph, people are born, somewhere around 10,000 bases, and they die of old age at about 5,000 bases. And telomeres generally get shorter as you get older. So the x-axis here is showing the age of a person, and the y-axis is showing the length of their telomeres. So when you go to get your telomere lengths measured at several places now you can do that. That's the normal line that they usually have. But a normal 50-year-old is going to see that where their telomere lengths are going to be right where that green dot is. They're going to have about telomeres a length of 6,000 to 7,000 bases in this case. But now, if your green dot turns out to be above there, that says that your biological age is less than your chronological age, which is good. And if it's below, your biological age is greater than your chronological age, which is bad. You don't want that. Now, if it's over here, you have accelerated aging. Here's where you have progeria. And then this is where everybody wants to be. So when I get to be 90 years old, I want my telomeres to be almost the length of a 24-year-old. And this is where we don't want to be, because that's actually dead. That's below the level of a progeric kid. So we want to keep from being there, but knowing the length of your telomeres is the first start, because there are things you can do right now to slow down the rate of telomere shortening. Now there's several different methods that exist right now for measuring telomeres. They're terminal restriction fragment analysis. That's actually the main type that I do at my company. Unfortunately, it's very expensive. It's not very quantitative. Well, you can make it quantitative, but it's more of a qualitative. You can say that one person has longer telomeres than another, or you can say that some telomerase inducer that you're working with has had an effect on the telomere length, but you can't really put a number on how big an effect that has been. Then there's also qPCR, which is just quantitative PCR. There's fluorescence in situ hybridization called FISH, FlowFish, HT-Q-FISH, and Universal Stella. These are all procedures that exist right now, mostly, some of them just in the research lab, some commercially, that you can use to measure telomeres. Now these bottom two do a new thing. That's looking at short telomeres, and I'll come back to that in a second. But the other ones really just look at the average telomere length. Okay, so when you take a human cell and you grow it in a petri dish, and you start looking at the number of PDL, the x-axis stands for population doubling level, so the number of cell divisions that it's gone through, you find a fairly straight line that when you measure the telomere lengths after different cell divisions, you find a fairly good straight line showing that as the cells divide, the telomeres get shorter. That's in line with what I was saying earlier. Now, occasionally, if you take blood from a person and measure blood from a whole bunch of different people, and then try to look at the age versus telomere length, sometimes you get a fairly straight line, but more often you get a very chaotic mess. And so telomere length measurement is not really in its prime yet. It's still a work in progress. And I think that there's a lot of, so there is a weak correlation. And why that is, I'm going to come back to, but well, I'm going to come back to it right now. Some people are born with different telomere lengths is one possible explanation for this variance. Rates of cell division between different people can occur. So remember, telomeres are a measure of the number of cell divisions. So if some person has more cell divisions, even though they're the same age than somebody else, then their telomeres are going to be a little shorter. Different environmental factors like smoking and stuff like that can also affect telomere length. And then what I think might be the main cause of a lot of this variability is that most of the techniques right now do have poor signal detectability. For instance, I know a lot of people that have taken had blood samples taken, and even though they have it tested 3 times in a row, they will get a pretty consistent number. But if they have another blood drawn, even the same day, and have that blood sample analyzed, they'll get different results. So there's still a few bugs that need to be worked out of this technique. So I think that right now, most of telomere length measurement is good for population studies. So you'll see a lot of publications where people have looked at like 1,000 different people and shown that there was a correlation between telomere length and some effect that they're looking at. But it's not really good for the individual. It's good for looking at the populations. But it is good for the individual to look at major differences. So for instance, if you were to test a patient of yours that you thought might be suffering from some type of accelerated aging-related disease, and you found that this person who is 60 years or 30 years old has telomeres of length of 60, I think these telomere length measurement protocols that exist today are good enough for doing that. Now, the abundance of short telomeres might be an exception. Okay, I've been looking at the short telomeres, a lot of publications, talking to people. I'm beginning to see that there is a stronger correlation between the abundance of short telomeres and health and aging than with the average telomere length. So average telomere length might really just be a good biomarker for the number of short telomeres that you have. And short telomeres might be, the big question is, why would the length of a telomere have anything to do with aging or health? And it might, there's a lot of theories about why that is, but the short telomeres, when they get really short, that might be the best explanation. If you use the shoelace analogy, where a chromosome is like a shoelace and the caps on the shoelace are like the telomeres, when that wears away, the shoelace starts to fall apart. When the caps get really short, the shoelace falls apart. That might be the same thing that's happening in our own cells. And so it might be more important to be looking at the abundance of short telomeres, not just the average to telomeres. This might a lot have to do with why the accuracy is better, is that when you look at short telomeres before and after some treatment like you see below, it's a lot easier to tell the difference between a short molecule and even a shorter molecule. Whereas when you start off with a long molecule, it's a little harder to tell the difference between a long molecule and a molecule that's just a little bit shorter, even though the length of the shortness is equivalent to what I showed before. So looking at the short telomeres might be a more accurate measurement than looking at average telomeres. But we don't know yet. There's still work underway that needs to be done, and so I'm keeping an eye on that. And, I'm thinking right now that short telomeres might be the way to go, or looking at both of them, both average telomere length and short telomeres. The next thing that we can do to manage our telomeres is to find a way to lengthen them. And that's where this enzyme that we talked about before, telomerase, comes in. This is showing telomerase. Well, first of all, I show this green thing as DNA, shown as a double helix here. And I'm showing telomerase here as a factory. And what telomerase actually does is it binds to the very end of the chromosome and then lengthens the telomeres. That's the function of telomerase. And as I mentioned before, we discovered this enzyme at Geron Corporation back in the mid-1990s. So let's go back to this bricklayer model, and I'll explain kind of what telomerase does here. The bricklayer is still going to fall off at the end of this wall in let's say, a reproductive cell, which naturally has telomerase. But like an angel, telomerase comes in and replaces that missing brick. So as a result, in our reproductive cells, when they divide, our telomeres don't get shorter. And that's why, for instance, our children are born with telomeres the same length as we have. Or they get reset during reproduction. But it's because of this enzyme telomerase. Okay, so evidence that telomerase is actually a useful tool for lengthening telomeres. I think one of the best examples of that came with Rhonda Pinnell, Dr. Rhonda Pinnell at Harvard, from a recent study he did with mice, where he used engineered mice and did work to lengthen their telomeres. And well, that was the publication. The press releases say that telomerase reverses aging process, where Rhonda Pinnell was actually, to his surprise even, able to reverse the aging in old mice. And what I want to do here is just show a news report from Diane Sawyer on this.
And now, eternal youth. Is it in a cage around the corner? News tonight of a breakthrough for some pioneering mice. But we always wonder, what does a fountain of youth for rodents reveal for humans? Here's Sharon Alfonsi reporting.
I feel tremendous.
In the movie Cocoon, it's a swimming pool that turns back the clock for a group of senior citizens. But now researchers have found a way not just to stop, but reverse the aging process. The key is something called a telomere. We all have them. They're the tips or caps of your chromosome, seen here in yellow. This is what it looks like in a young adult. But as you grow older, the telomeres become damaged and frayed, and as they stop working, we start aging, experiencing things like hearing and memory loss. Scientists took mice who were prematurely aged, added an enzyme, and essentially turned their telomeres back on. You can see it before the enzyme, after.
Their brain function improved, their fertility was restored. It was a remarkable reversal of the aging process.
Look at this picture. The mouse on the right has bad skin, gray hair, and is balding. But the one on the left had its telomeres flipped back on.
And you could see that essentially you now have a dark coat color, that the hair is restored, that the coat has a nice healthy sheen to it.
Even more dramatic, the change in brain size. This is before. The mice had 75% of a normal brain, like a patient with severe Alzheimer's. But after the telomeres were reactivated, the brain returns to normal size. As for humans, while it is just one factor, scientists now say by looking at our blood cells and measuring those telomeres, you can get a better idea of how well you'll age. The longer the telomere, the better the chances for a more graceful aging. But as for tinkering with them and turning back our aging process, researchers say we still have a long way to go. Sharon Alfonsi, ABC News, New York.
Well, I think that's pretty exciting. I think it's the best proof of concept that we've ever seen for the fact that telomerase and lengthening telomeres could actually have an effect of reversing aging. And it kind of fulfills a dream that a lot of us in this room have had for 20 or more years, that could we actually succeed at doing this? And this is the first proof of concept. Now, even more recently, Maria Blasko, who's been mentioned a few times already, she just published a paper where she used gene therapy to produce telomerase inside of mice, and actually found that when she did that, she was able to extend the average life. Let's see if I can remember how you just showed me to do this. Let's see. The average lifespan of the mice was increased 24%. This is a younger group of mice. And the maximum lifespan was increased 13% by producing telomerase by gene therapy into mice. Again, older mice showed a 13% increase in the average lifespan, and even a bigger increase, 20% increase in the maximum lifespan. So again, this is kind of like proofs of concepts that are fulfilling the dreams that a lot of us have had and been working very hard to pursue for years and years and years. So this is, again, very exciting stuff. So this is where it is right now, but what I want to do is talk a little bit about what my company right now is doing. I'm not going to spend a lot of time on this, but I'm just going to say how we are searching for telomerase inducers. And this is the same way that the telomerase inducers that exist already were found. And that is based on the fact that telomerase is produced by the telomerase gene. You know, all of our cells have genes. Same thing, give us blue hair, blue eyes or blonde hair. There's a gene for telomerase, and it's producing telomerase. Now, that's true in our reproductive cells, where our telomeres don't shorten. But in all the other cells of our body, there is a repressor that binds to a regulatory element, kind of an on-off switch next to the gene, that shuts that gene off. So in all of our cells, there is something, we don't know what that protein is, but it's called a repressor. We don't know what it is, but it shuts off the gene somehow. And so the goal of what Geron Corporation did, other corporations did, and Sierra Sciences is doing, is looking for a drug that would dislodge that repressor and just allow the gene to turn back on again. And if you want to turn it back off, you just quit taking the drug, because every drug like aspirin or anything else has a half-life that can dissipate if you quit taking it, and you can shut the gene back off again just by letting that repressor rebound. And so that's the way the research is going right now.